First, it's important to understand a few things about the life cycle of the hermaphrodite. She has about a 3-day life cycle. After an embryo is laid on the plate, it takes about a half a day for that embryo to hatch and become a first larval stage larva. From there, it takes about a day and a half to reach the L4 larval stage. And this is the last larval stage. From there, this is the last larval stage where we know that the worm is not yet producing its own offspring if it's hermaphrodite. It takes one more day to reach adulthood. In other words, the worms have an approximately 3-day life cycle. This is important to know because it means that if you leave an adult hermaphrodite on a plate with its own children, within 3 days, that adult will also have its adult children on the plate, and that makes it harder to tell which adult is the one that you're trying to count the offspring of and which are its offspring. If we diagram a plate as a circle, and a focal worm as a little squiggle, in this case a black squiggle, we're going to call that the P0 or parental generation worm. Now if that's that one plate on day 1, so we're going to diagram time moving that direction to the right, and if we have plate, we'll letter the plates. Plate A, the first plate you put that adult worm on. If you start with an adult, then when you look at that worm's plate on day 2, same plate, that adult will still be there, and it will have produced some embryos. So we're going to indicate that red indicates the F1 generation. That's the generation of worms that you are trying to measure, count, the total number of offspring of that worm. Now remember, if you start on day 1 with an adult, one problem could be that in its past, before day 1, if it was an adult the previous day, for example, it would have already produced some of those F1 offspring on a plate that you didn't count, which means that you haven't had the opportunity now to count the total number of offspring that worm produces. That means that we need to start instead on a separate plate, let's say. What you want to do is start with, ideally, an L4, that's the last larval stage, larva. Then on day 2, it would have become an adult and probably started laying its first embryos, depositing the embryos on the plate. Those are its F1 kids, and those are the ones you're trying to count. Now back to plate A. If you leave that same worm on the same plate, what's going to happen? It's still an adult on day 3. Day 3, it's still laying, producing more self-offspring as embryos. But the previous day's embryos have now become larvae. So still the same generation, and again, these are all individuals that you want to be able to count, both the larvae and the embryos. So if you keep that adult on the plate, by the 4th day or so, its F1 oldest offspring, the ones it lay first, are now about adults, which means it's starting to get really hard to know which are the offspring, because what the problem is that once those F1 offspring become adults, they start producing embryos of their own. Those would be the F2 generation, the grandkids of the worm you're trying to count the fecundity of. And then things get complicated, because if you leave that same worm on the same plate for multiple days in a row, in other words, it becomes really difficult to tell which offspring are you counting. Are you counting only the red generation, or are you starting to count some of the F2 generation as well? And we know that, or I'm going to tell you right now, we do know that a single adult will produce offspring over perhaps 5 to 7 days, which, as we saw in the prior slide, is about 2 whole generations of worms. They've got about a 3.5 day life cycle. So a single adult will produce two generations, in 2 generations will produce all of its offspring. So you can't, in other words, here's the take-home message, leave the adult on one plate for its entire life. It becomes impossible to track which worms you're trying to count. So there's a solution to this problem. The solution just requires a little bit more work and a few more plates. So again, across the top, we'll diagram time, with each column being a different day, so time in days. And each row of plates, again, is going to be a different plate over time. So plate A, plate B, plate C, plate D, and so forth. So remember, we want to start, for every single worm you want to measure the brood size of, this is going to be what happens. And because you need replication, you'll do this for maybe 3 or 5 or more individual worms that you're trying to measure the fecundity of. So, but for a single worm, starting as a larva, hopefully L4, but any larva would do, the closer to adulthood the better to start the experiment. L4, day 1. On that same plate on day 2, as we just saw, that worm will be an adult and maybe started to produce some embryos. Now, here is what you need to do for every single adult that you're trying to measure the brood size of. Day 2, you transfer that worm to a new plate. What does this do? That means on day 3 when you come in, that still adult worm has produced more of its self-offspring on a second plate. Now let's go back to plate A. In that 1 day's worth of time, the initial offspring, the F1s, have hatched and become little larvae. And then for them, the next day they're probably L4 larvae, and the day after that, by day 5, all 4 of those initial offspring are now adults, and they, as we just looked at, will start producing embryos of the next generation, the F2. So the same thing happens on the second plate. Those embryos that were embryos produced on day 3 become little tiny larvae by day 4, L4s by day 5, and adults by day 6. So you keep moving that hermaphrodite every day to a new plate. So it spends 24 hours on a plate producing offspring, and then it gets moved again. Produces offspring. Day 5, you move it to plate E. And what we're looking for is that at some point, day 6, you move it to plate F. At some point you're going to come in and see that while every previous day that worm had produced some embryos in that 24-hour span it was on the plate, one day you'll come in and there will be no embryos on the plate. So in a 24-hour span, in other words, no offspring. That is the signal that tells us that that hermaphrodite has finally run out of self-sperm and is done fertilizing its own embryos, its own oocytes. So that's what we're looking for. That signals that we can stop moving that worm onto more and more new plates. So, how do we now count all of these offspring? Well, you could consider counting the offspring on day 2 as little tiny, plate A, day 2, little tiny embryos. But those embryos are really hard to see under the microscope. So you could count the 24 hours worth of worms on day 3, but why not wait until day 5 when all of those 4 initial embryos produced between days 1 and 2 on plate A are now adults. They're huge, they're moving around, they're easy to see under the microscope. So that is basically the trick, is because these worms have an approximately 3-day life cycle, and those worms are produced between days 2 and 3, you wait until day 5 for plate A to count. And then you count the offspring from plate B on day 6 of your experiment, and so forth. And so those final offspring that were produced on plate E in this example were then larvae on day 7, maybe L4s on day 8, and finally adults, and easy to count by day 9. And because this hermaphrodite was sperm depleted by day 6 or 7, there were no offspring on that plate, there's nothing else to count. So then you simply sum up the total number of worms on all, in this case, 5 of those plates, and that tells you the total number of offspring that hermaphrodite produced. That's the measure of its individual self-fecundity. So in general, every worm you work with will probably take around 7 NGM plates, and each round of counting will take probably around 10 days. So here are another couple of pointers. Indeed, by the time that those F1s, the red worms, the offspring, are adults, they're going to have started producing F2s, their own kids. So how do you avoid counting them? Well, that's why it's important to count as soon as that plate becomes adult, because it's really hard to see those embryos or even L1 stage larvae with our microscopes. So on day 5, there will be 2 generations of worms on this plate, the kids and the grandkids. You only want to count the kids, the red worms, but you won't really be able to see the grandkids, the blue embryos or tiny larvae at this stage. Now, if you waited another day or 2, if you counted plate A on day 6 or 7, then the grandkids are starting to get into adult-sized territory too, and that would make it impossible to get an accurate count. So you do have a maybe 1 to 2 day window around day 5 for plate A to count reasonably accurately the offspring and not accidentally count the grandchildren as well. Now, I've indicated that you should probably move the worms every day, the focal worm, from one plate to the next. But that doesn't work very well on weekends, for example. And so there are a couple of things you can do. It is feasible, because it's a 3-day life cycle, you could move that focal worm every other day to a new plate. That would still put enough time between generations that you could still distinguish the parent from their offspring at the end of those 2 days. So that's one approach. For a weekend, what's best to do is to move the worm on, say, a Friday late, maybe Friday afternoon, and then come in for plate 3 Monday morning, or at least as early as possible when the lab's available on Monday. So that's about a 2.5 day period, and because again it's about a 3-day life cycle, you should still be able to distinguish the adult from its kids, even though they'll be almost adult between, if they were embryos on Friday afternoon, the offspring would have grown to late L4 young adult size by Monday morning. You can still distinguish the adult, the parent, from its kids at that point. So that's one approach to solving the issue of what happens on weekends. The other thing you can do, especially if you're not doing an experiment that controls temperature, is you can also move the worms that you're studying into a 16-degree incubator instead of the 20-degree incubator we usually grow the worms in. At a cooler temperature, the worms will grow more slowly, and that buys you a little bit more time to reduce the speed of their growth and make it easier to distinguish the parents from the kids on Monday morning. And now, I don't expect that you will have this sort of control over the process, but what really might be ideal in terms of which days of the week to do these steps of the experiment could be Thursday, plate a worm. Then Friday, Saturday, Sunday, Monday, 16 degrees. And that puts the bulk of the worm counting, those green plates, on weekdays. And you should be done or about done by sperm depletion by the following weekend. And if necessary, then you could also grow that adult worm again at 16 degrees over those last maybe day or 2 of its self-fecundity and get the final brood size count the following Monday. And now I just want to leave you with a couple of tips about how to count the offspring on these plates in, hopefully, this 1 week of counting the offspring on a bunch of plates. There are a couple of tips to make that process a little bit more efficient. So the difficulty can be you've got a plate with maybe 40 or 60 or 80 adult worms on it. How do you make sure that you don't double count any of those worms as they're crawling around the plate? So there are 3 different tips. One is to keep track of where on the plate you've already looked. I like to draw a simple black grid on the lid of a separate plate. And then I flip that lid upside down and place it underneath the petri dish that contains the worms. So there's the OP50 lawn, there are worms on there, but there are also some worms off the lawn, and a lot around the edge of the lawn. So when I'm looking at this plate under the microscope, I start in one quadrant, count the worms in that square, well, not quite square, at the edge of the plate, then that square, that square, that square, move back and forth, and if you do this relatively quickly, you make sure that you only count each worm once. One worm won't crawl from sector to sector if you're quick enough. The next thing to do to make sure that you don't really have to speed through this process is put the plates in a refrigerator, not a freezer, a refrigerator, 4 degrees, for maybe 15 to 30 minutes before you count. The cold simply anesthetizes the worms. It makes them stop moving, and they stay cool long enough after you take them out of the refrigerator to get a pretty accurate count. The last tip, as you can see with this photograph in the upper right, is I have a couple of manual tally counters where every time you press down on the button it advances the counter 1. So instead of trying to memorize how many worms you've looked at while you're staring into a microscope and moving the plate back and forth, you just press down on the button every time you see a worm. This is probably more accurate than trying to remember the total number of worms you count per plate. And then when you're done with that, you can always use the other button to count another plate of worms and then twist the knob on the left side to reset the counter for your next plates. So here's the summary. Make sure you start with a larval hermaphrodite. If you start counting with an adult who's already produced offspring, you haven't made an accurate count of fecundity. Make sure each of those worms is singled onto its own plate so that you're counting just the offspring of one worm. Move that worm every 1 or 2 days, except for weekends when it would be Friday to Monday probably. Really critically, because you're going to be doing replication, you may have 10 different worms that you're individually moving day to day. So you need to assign each worm a name. You might call the worms worm A, worm B, worm C, and label the plates accordingly so you know which plates worm A was on day 1, worm A on the second day, worm A on the third day, the same for worm B, and so forth. Because you have to tally some, that is, only the offspring for worm A, that's its individual lifetime brood size, separately from all of the plates that worm B was on, and so forth. Ask your instructor to help you, well, the instructor will do it for you. Reduce the incubator in the lab to 16 degrees in case you want to cool down and slow down growth of your worms over the weekend. 3 days after the mom, the parent hermaphrodite, was removed from each plate, then count their F1. They'll be adults 3 days after. As we just discussed, cool those plates before you count worms, and consider using a tally counter and that transparent grid under the plate. Happy counting!